I will have some overlap with the previous presentation, but I think may not be bad to kind of have this repetition to really understand what we are doing. Yeah, I would like to present actually a real case study from the cardiovascular area where we employed graphical procedures to multiple testing according to the presentation from Bretz, Maurer, Brannert, and Posch in Statistics in Medicine 2009. And before I start, really, I would like to acknowledge contributions from Frank and Willi Maurer is here as well, and Bjoern Holzhauer to this presentation. So let me try to work this. So I have to go page down. Okay, good. So I will start with a brief introduction to the graphical approaches to multiple testing, and then I will briefly go into a case study where the graphical approach helped tremendously to interact with the project team to arrive at an informed decision about the multiple testing procedure that we chose for this case study. So why do we think graphical procedures are useful? At least in the cardiovascular area, we witnessed that confirmatory outcome trials have become more and more complex with the need to account for multiplicity for various sources like multiple treatment arms, multiple primary and secondary endpoints, and interim analyses. So designing a valid multiple testing strategy with desired properties needs to take into account the needs of many stakeholders that may look at the study from different perspectives and have different needs. So that is a cross-functional exercise and may involve several iterations, as we heard. You kind of design a draft procedure, and then you look at the properties, and then you refine it, look at the properties again, and so on. And throughout this process, you have a close interaction with the stakeholders who are non-statisticians. And for that reason, it is very important to be able, first of all, to translate the clinical considerations or the project team's considerations into a testing procedure, and then also to be able to report it back in a transparent and understandable way. And that is the only way how you can then reach at a decision or testing procedure that really reflects the needs of the stakeholders. So to this end, the graphical approaches provide a framework to tailor advanced multiple test procedures to structured families of hypotheses. We just heard already how those are defined. They are very helpful to visualize complex decision strategies in an efficient and easily communicable way. And by just following a few very simple rules, they ensure strong type 1 error rate control. So just very quickly, notation. I think this is all nothing special. Null hypotheses are denoted by H1 to HK. We have initial allocation of the significance level among those K hypotheses, and we have unadjusted p-values denoted by P1 to Pk. So if a hypothesis Hi can be rejected at level alpha I, that means if the unadjusted p-value is less than alpha I, we reallocate this local level alpha I to the remaining not yet rejected hypothesis according to a prefixed rule, and continue then the testing with the resulting alpha levels. And this is called alpha propagation. So let me briefly introduce a few simple conventions for the graphical procedures. Hypotheses are represented as nodes. Here 2 examples, H1, H2. We split the significance level alpha, and we This is denoted by the values above the nodes or below the nodes, however you want to do it. And alpha propagation is indicated through weighted directed edges, and the weights determine the fraction of the local alpha level that is passed on to the other hypothesis. The weights are between 0 and 1, and the sum of the weights of outgoing edges from a node must not exceed 1. So these are very simple rules. And now briefly to our case study. So the case study is a randomized, double-blind, event-driven outcome trial in stable post-myocardial infarction patients. We study 3 doses of a new therapy versus placebo on top of standard of care. The reason we are doing that is that there was no surrogate available for dose finding, so we had to carry multiple doses into phase 3. Primary endpoint is standard endpoint in this area, a composite of CV death, MI, or stroke, analyzed with time to first event methodology. And we have 2 key secondary endpoints targeting additional label claims, and that is also based on request by FDA. So we had to include them in the multiple testing procedure. Yeah, so the first endpoint is an extended composite endpoint where we add unstable angina requiring urgent unplanned revascularizations to the composite. And the second one is an analysis of a subgroup of patients with prediabetes where we look at time to new onset type 2 diabetes. And additional multiplicity is introduced by 2 planned interim analyses. So some clinical considerations are given on this slide. The one is that primary endpoint is essential to establish efficacy of the respective dose group, and the key secondary objectives target additional label claims for doses that have already established efficacy based on the primary endpoint. So that introduces a concept that's called successiveness, that we do not want to reject a secondary hypothesis for a dose without having rejected the associated primary hypothesis. Another consideration is that higher doses may potentially be more efficacious, and potentially is maybe is stressed here, but we are pretty sure that lower doses are generally safer. So what we would like the testing procedure to do is to allow testing of lower doses regardless of the efficacy of higher doses. And also we want to split the alpha unequally. That means that for the highest dose, we think we need larger effect sizes to compensate for the expected worse safety. So this gives us a 4-armed trial, 3 doses versus placebo, as I said, and 3 endpoints. And in total, we have then 9 hypotheses with a certain structure induced by the combination of 3 doses and 3 endpoints per dose, among which we would like to control the family-wise type 1 error rate in the strong sense. So let's now see how a multiple testing procedure can be designed to address these clinical considerations and needs using the graphical approach. So first, the hypotheses related to the primary endpoint are represented as nodes. And we have 3 doses, so we have 3 primary hypotheses, H1 to H3. Then we represent the hypotheses related to the secondary endpoints that are incorporated in the multiple testing procedure. And the nodes for the secondary endpoints represent families of 2 null hypotheses each related to the 2 key secondary endpoints for each dose. So So secondary endpoints, we said, are of interest only if the study is positive and if the primary hypothesis for the respective dose is positive. Therefore, the initial alpha allocation is 0. So the complete alpha is initially allocated to the 3 null hypotheses for the primary endpoints. And as I said before, we have increased the hurdle for the highest dose because of the expected safety profile. So we want to have relative better efficacy to result in an acceptable risk-benefit profile. So if the null hypothesis related to the primary endpoint is rejected for a certain dose, we would like to propagate alpha, a portion of alpha, to the primary objective for lower doses, because we think even if a high dose has been shown effective, it is of interest for risk-benefit considerations to also have a lower dose successful. So this is represented in the graph through the directional edges from high dose to medium and low dose, and from medium dose to low dose. So we would also like to propagate a proportion of the alpha to the corresponding secondary hypothesis, and this is represented by the directed edges downwards. And the next thing to decide is the weights of the directional edges. That means what proportion of alpha is propagated to which node, with the restriction that the sum of the outgoing edges have to be 1. So if the primary objective for the high dose or medium dose is rejected, we consider it relatively more important to increase the likelihood to reject the primary hypothesis of a lower dose than to test the secondary objectives at the same dose. So we have kind of a weight of 0.7 to 0.3 among those two choices. So actually, we propagate 0.45 from the high dose primary objective to the medium dose primary objective, and 0.25 to the low dose primary objective, and 0.7 from the medium dose primary objective to the low dose primary objective. And if all hypotheses related to a secondary objective for a dose is rejected, we can also propagate the local alpha, and we do that to the primary endpoint for the next lower dose. Except for the lowest dose, we propagate the alpha to the primary endpoint of the medium dose. And as I said, the key secondary endpoints within each dose group will be tested with a weighted Bonferroni-Holm test at the available local significance level. And that could be easily represented in the graph, but for this presentation, I chose to simplify it to illustrate the method better. And next point I would like to say, I mean, we heard that before, the graphs are based on weighted Bonferroni type tests, and this multiple testing procedure that is represented by this graph can be improved by exploiting the correlations between the 3 primary hypotheses, H1, H2, H3, that share a control group. So there is a correlation that we can use. And the improvement is that we use the weighted Dunnett tests for all intersection hypotheses that contain at least 2 of these 3 primary hypotheses. And this testing procedure can still be represented as a graph, at least the way how the alpha is propagated. And yeah, I want to leave it as like this for now, but in the backups, if the slides are distributed, we have the full graph for this testing procedure. So let's now illustrate quickly with an example rejection sequence how the sequentially rejective multiple testing procedure defined by this graph actually works. So we assume that we can first reject the first hypothesis related to the high dose versus placebo comparison at the local 0.2 alpha level. And from the directional edges, you see that in this case, 0.25 times 0.2 alpha is then propagated to H3, 0.45 times 0.2 alpha is propagated to H2, and 0.33 times 0.2 alpha is propagated to H4. And that is reflected in the updated graph. And you see now we have also alpha allocated to the secondary endpoints for this high dose, and we increased the alphas for the medium and low dose versus placebo comparisons. So next we assume we reject the family of hypothesis H4, and from this you see we will propagate the 0.06 alpha to the hypothesis H2, which is reflected in this graph. and next we assume that we can reject H2, and I think you got the picture. Then we can again update the graph, and next we no, it is already the end of the rejection sequence. Now, okay, I'm back here. And now you see that we have almost full alpha for the low dose versus placebo comparison. So yeah, I think it's important to note that the conclusions from this sequentially rejective multiple testing procedure is really independent on where you start to work through the graph. So whether you first reject H1 or first reject H2 doesn't matter. You always arrive at the same conclusions, and you will be sure that by setting it up like this, that you ensure strong control of the type 1 error. So let me just comment on interim analysis. So how do we deal with these two planned interim analyses at 50% and 75% information fraction? Yeah, we decided to just use a Bonferroni split across the times, so that we assign very small alpha, 0.01% for the first interim analysis and 0.04% for the second interim analysis. That leaves 2.45% for the final analysis. And then at each time point, we can fully exploit the correlations between the primary test statistics. And yeah, things would be very messy if you also would kind of try to do alpha spending across the time points and try to exploit also the correlations among the time points. So in summary, confirmatory clinical trials are becoming increasingly more complex, often comparing multiple doses or treatments with a control for several primary and secondary endpoints. Multiple study objectives can be reflected by structured families of hypotheses, usually that are characterized by multiple groups of parent primary hypotheses and descendant secondary hypotheses. And the novel graphical approaches for constructing and visualizing complex multiple test procedures with a focus on structured families of hypotheses are well suited to facilitate the communication in clinical teams and to provide transparent decision strategies. So these procedures ensure strong control of the overall type 1 error rate across all primary and secondary endpoints. And of course, they have to be customized, not only based on the clinical considerations, but also based on the operating characteristics obtained via clinical scenario simulation. And yeah, we think how EMA could help, or in general, would be nice to have kind of harmonized terminology and framework characterizing study objectives and endpoints with respect to the impact on approval and labeling, and also with respect to the need for type 1 error control for the different levels of impact on approval and labeling. So here are just a few references, and the last one actually has a link to a publicly available program package in R that can be used to implement these graphical procedures. So thank you for your attention.